identification of novel molecular markers for early and serious rheumatoid arthritis. So if you have questions, please don't hesitate to ask them. Maybe I'll start with an introduction on rheumatoid arthritis. Rheumatoid arthritis is an autoimmune disease characterized by chronic inflammation of the joints. So it's actually the most common autoimmune disease with a prevalence of 1%. So 1 in 100 people will be affected with the disease. We still don't know at this moment what is the cause, the etiology, and which target antigens are actually affected in the disease. So what's actually happening in patients with rheumatoid arthritis, or when you compare it with the normal joint? So here you see actually a picture of the normal joint. So we have the bone with the cartilage over here, and this will be encapsulated with a joint synovium. This is a tissue with specific antigens present over here, and this will be encapsulated with a joint capsule. So this is just a normal situation. What happens now in a patient or a joint affected by rheumatoid arthritis, we really see erosion of the bone and the cartilage over here. What is really characteristic of patients with rheumatoid arthritis is that they really have an inflamed joint synovium. So there really are a lot of inflammatory processes going on in this inflamed synovium. Together with this inflamed joint synovium, we also see swollen joint capsules. So you really see that this capsule is really very thickened over here. So what is the really characteristic? There are a lot of inflammatory processes going on in this joint, and a number of proteins are added over here. And what is actually the cause of the disease? I mean, we really don't know the exact cause, but we really know that the patients develop an immune response against their own joints over here. This is characteristics of patients with rheumatoid arthritis. So what actually are we doing? So we really, currently there really is a need for reliable disease markers in autoimmunity research, not only for rheumatoid arthritis patients, but also patients with, for instance, inflammatory bowel disease or other autoimmune diseases. I think there is definitely a need for reliable disease markers. So why do we need these markers? I think for a number of patients with autoimmune diseases, we still don't have good markers that we can use for the diagnosis of these patients. So I think it's really important if we have good laboratory markers that can measure a single molecule, for instance, or a protein in the blood, that can really help us to diagnose these patients. I think this is really a step forward, I should say. Another application where we really need to use these biomarkers is the prognostic potential. So if you speak to doctors, I think it's really important to know that the doctors actually say, if I have a marker which actually predicts that a patient will suffer from the disease much more than another patient, that really helps me a lot. So if I have this marker in this patient when he presents at the first visit, and when I can test for a certain marker, and when this marker says something about the prognosis, that really helps me a lot. Also, another application of the markers is the subtyping. So the subtyping according to their pathogenic profile. So what does this actually mean? I think if you look at autoimmune diseases in general, autoimmune diseases are actually a group of diseases. So if you look at, for instance, also rheumatoid arthritis patients, it's not one single disease. So it's actually a group of many different patients which have different characteristics. And therefore, we really need to have specific markers that we can actually use for the subtyping of these patients. So that's another application. So if you have good markers, then of course, we would also like to use these markers for the development of specific therapies. If you have a specific marker that's, for instance, overexpressed, or then you really want to have therapy that really can down-regulate this marker. Theranostic application, that's a fancy name. What does that actually mean? Is that your marker predicts something about the interference of a medication. So if your marker is actually changing by interfering with the therapy, then this actually means that this has a theranostic application. And finally, I think really as basic researchers, we also really want to have new biomarkers to know some more information regarding the disease pathogenesis. That's an important thing, I should say. So if you now look at the diagnosis of rheumatoid arthritis, patients with rheumatoid arthritis are actually classified according to the criteria of the American College of Rheumatology. So these criteria actually entail both clinical parameters, so stiffness of the joints, etc. But what is really important, that these criteria also contain testing for 2 serological markers. And what are these markers? These 2 markers that these patients will be tested for. The first one is rheumatoid factor, or RF in short. And what is rheumatoid factors? That means actually that there are antibodies present in the serum or the plasma of these patients that are directed against an Fc portion of immunoglobulin G. So what is rheumatoid factor? It's a factor that has been used for a long time in the clinic already, but it's not an ideal marker because it has a sensitivity of 70% and it has a low specificity. So what does it mean? There are also patients with some other rheumatic diseases, but not rheumatoid arthritis, that will also test positive for this rheumatoid factor. So that means that the specificity is not really high. Then recently, actually, novel marker was added to the criteria of the American College of Rheumatology. And this was actually the presence or testing for the antibodies against citrullinated proteins. So what are citrullinated proteins? That means that arginines in proteins are actually mutated or modified to citrulline amino acids. So this is called citrullinated antigens. And this is really specific for patients with for instance, autoimmune diseases, but more in particular patients with rheumatoid arthritis. So these patients express citrullinated proteins in their synovium, for instance. And we don't find these citrullinated proteins in the normal situation. So what is then actually more characteristics is that patients with rheumatoid arthritis develop antibodies against the citrullinated proteins. And these markers can be used also as a serological marker for the diagnosis of rheumatoid arthritis. So the sensitivity is approximately 70%, but what's really important for these markers is, of course, that it's really specific for the disease. So these 2 serological markers are really important for the diagnosis of rheumatoid arthritis. But if we look at the whole group of patients with rheumatoid arthritis, then we see that the prevalence of rheumatoid factor and ACPA, that's the short for antibodies against citrullinated proteins, the prevalence is much lower in patients with early disease. And this is actually the most important stage because the patients visit their doctor and then we really want to know, do I have rheumatoid arthritis or not? So we really need markers that can say or that are valuable in the early phase of the disease. What's also really important, that 30% of the patients with rheumatoid arthritis, they remain negative for both or either rheumatoid factor or ACPA. So that is what I just mentioned. Rheumatoid arthritis is also a group of disease, and we have a mixture of different patients. And it seems just that approximately 1/3 of the patients will not test positive for the current markers that are being used in the clinic. So this is just an introduction on the diagnosis of rheumatoid arthritis. So what is then actually the main aim of our study? We really want to have additional markers, and especially for patients with an early disease and patients that are both negative, seronegative, that's what we call, for both rheumatoid factor and ACPA. So our aim of our study was, of course, that we would like to identify novel disease markers for, on the one hand, patients with an early RA disease, and on the other hand, patients that were seronegative for both serological markers that are being used in the clinic. So how do we do this? We are using a methodology which we call serological antigen selection, or short SAS. And this procedure is actually based on the analysis of all the antibodies that are present in RA serum or plasma to identify the real target antigen. So we really want to see which antibodies are present in the patient's serum and to which target proteins are they directed in, for instance, synovial tissue of patients with rheumatoid arthritis, because we know that the synovium is really affected in these patients. And then the SAS technology is actually based on cDNA phage display, and I really would briefly like to remind you to the use of phages. Phage is, of course, a virus that infects bacteria, and here you see a cartoon of the phages that we are using in the laboratory. These are filamentous phages. They have a lot of fold proteins. What you see here on the one hand, P3, we have P6, P8, P7, and P9 indicated over here. And this is just an EM picture of these filamentous phages. But what is the main characteristics of these phage viruses is that we can use this as a cloning vehicle. So that means that you can actually clone. We have, of course, DNA in the phage genome. So we have a phage virus with DNA, which is encapsulated with the proteins, what you see over here, which corresponds to this over here. And what is now the characteristics? That we can actually insert foreign DNA into the genome of these filamentous phages, and of course, upon expression, after insertion of the cDNA or DNA in this case, you will have the displayed protein on the tip of the filamentous phages. So that's actually the basic principle of the SAS technology that we're using. So we're cloning a lot of cDNA fragments in the genome of the filamentous phages. When the phages are expressed, they display the protein corresponding to the cDNA that you have cloned in the genome of the phage. So there's a direct link between the genotype and the phenotype for the expression. Is that clear? So, of course, the power of the technology is not that you can clone one single cDNA fragment, but what you can do is, of course, you can clone multiple genes into the filamentous phages over here in order to generate these huge libraries. So what you, at the end, you can do, you can actually create these huge libraries with over 40 billion different fragments over here that will be actually expressed on the tip of the filamentous phages. So that's actually the basis of our SAS procedure. So how do we now use this SAS procedure in order to find novel biomarkers for rheumatoid arthritis? So the first thing that we need to do is to construct a well-defined RA phage display library. So how do we start? We actually start to collect RA synovial tissue. So we collect tissue from an RA patient, so you know that this tissue is affected, it's inflamed, there are a lot of complexes going on in this synovium. So we collect the tissue from this patient. The next step is, of course, that we first isolate the mRNA out of the tissue, then convert it to the cDNA, and the next step is, of course, that we insert the cDNA into the genome of the phages. So that, of course, when we express the phages, at the end, we have the displayed protein, which is actually expressed on the tip of the filamentous phages. So the final goal is, of course, that we clone all the cDNA fragments that were present or originating from the RA synovial tissue that we will actually mimic in a test tube in the laboratory so that we actually create an RA display library. So that's the basic principle. So we really need to do a lot of quality controls for this RA display library because this is the start of our experiments. We need to have a very high number of protein fragments present in order to find novel biomarkers for rheumatoid arthritis. So how do we construct the cDNA basis library? This is actually done via fusion of the phage code protein B6. I already mentioned it on one of the slides that I showed you. So what do? We have a characteristic of one of the phage code proteins, which is called protein 6. And this protein allows the fusion of to its C-terminus, and therefore it makes it ideal for cDNA display. So how does it actually work? So this is just our vector. We have our sequence of the gene 6. Then we have a linkage sequence that we use, and then we clone our cDNA, and in this case it's a cDNA that we have originated from the RA synovium. We just clone this as a fusion to this minor code protein G6. So when we express the phages over here you see it more in detail. We have gene 6, the linker here, and then the cDNA. And then when upon expression, the cDNA will be expressed as a fusion to the gene 6 protein. So that's the plan. So what do we have in our laboratory? We have vectors available for cDNA display. And what is really important, if you would like to express proteins, it's really important that you need to express the proteins in the correct reading frame. So therefore, we actually clone our cDNA that we actually isolate from RA synovium in 3 different phage display vectors that correspond to 3 different reading frames. And the final aim is that we have at least one cDNA expressed in the correct reading frame. So that's the step. Here you see a more detailed information about the construction of these P6 phage display vectors. So this is what I just showed you. And we start with, of course, a Laxat promoter in our vector. We have the gene 6, then we have the glycine serine linker, the multiple cloning sites where we are going to clone our cDNA, and then we have the stop codon. And this is actually the same thing, but more detailed information. So we start with gene 6. So here you see the DNA sequence. Then we have the linker sequence, which corresponds to this. Then we have the multiple cloning site. And what you see on these 3 different lines is given PSP A, B, and C. So these are the 3 phage display vectors. And what is the only difference is that they just differ on one single nucleotide. And if you clone then your cDNA fragments in all the 3 different vectors separately, then you actually end up with the cDNA library expressed in reading frame 1, reading frame 2, and reading frame 3. So that's actually the plan. So you have, in total, you have 3 different libraries. The RNA library in PSP A, B, C, so which corresponds to the cDNA fragments that are cloned in 3 different reading frames. So if you then combine this, you will have at least one cDNA that will be expressed in the correct reading frame upon expression of the filamentous phages. Is that clear? So, more in detail, how are we going to clone the cDNA library into P6 vectors? Because it really seems very straightforward to do, but I think it's really important that we just need to prepare cDNA or DNA from a library. The library can be just originating from the tissue, but that can also be an existing library that is already present in a plasmid vector. So what we most of the time do is when we have made a cDNA library over here, we first actually express, this is just the main outline of the creation of the display library, but most of the time we already have some libraries available in other Lambda systems, so which are not displayed on filamentous phages. So what we can do is then we have our cDNA fragments that are cloned in a plasmid vector. And this is one of the ways you can go. So what you can do is when you have your cDNA library present in a plasmid vector, then you can prepare DNA from the cDNA library in the plasmid vector and then digest it with the different restriction enzymes. So how are we going to do this? This is just an example. So this is all DNA from the plasmid vector, which contain all the cDNA fragments originating from our RA synovial tissue. So when you then digest your plasmid vector, you see a number of cDNA fragments. So these smears over here actually correspond to all the different cDNA fragments that are present in the library. So, and what do we need to do in order to clone these cDNA fragments from one plasmid vector to another, to the phage display vectors? We just purify all the fragments, actually initiating from cDNA fragments from the size of 500 base pairs to the vector size. And the vector size is here normally, we know in general, the vector size is 2.5 kb or something. So we just isolate all the cDNA fragments and we then ligate these fragments into our 3 phase display vectors in the 3 different reading frames, PSP A, B, and C. And what's also important that we also need to cleave these vectors in order to do the ligation. So if we've not cleaved or digested our phase display vectors, we cannot do the ligation. That's clear. Yeah? So if you have done this ligation, so all the purified fragments, ligated these into digested phase display vectors, which are also digested with the same restriction enzymes as we've digested the cDNA fragments, we can then transform the ligation mixtures in electrocompetent bacterial cells. So here we are using E. coli TG1 cells by electroporation. And we did this in the laboratory, so we had actually 3 different libraries that we generated. So the RA library in reading frame 1, so corresponding to PSP6A. We had B and C, and what was really important to know is that we had at least 1 to the 6 colony forming units, because this was our criterion. We really want to have high diversity libraries. That means we need to have a high diversity of all the different cDNA fragments that were present in the synovial tissue initially, and we really want to clone these into these phase display vectors. So if you then just add these 3 different libraries together, then you have a total library size into the phase display vectors of 1.1 times 10 to the 7 different colonies. So you have at least 7 million different colonies that you generate, corresponding to a number of cDNA fragments. So questions on the construction of the library? Because I don't know whether you are familiar with the cloning, because this is...
Can you use electroporation?
Yeah.
Yeah,
of course there are many different technologies to do that. Of course for bacterial cells, we most of the time use heat transformation or just by an electrical pulse. And some bacterial cells are more are better performing doing a heat transformation than by doing an electrical pulse. And that's why we did this. Yeah, it's just a preference.
Yeah, I was just wondering because you did digest factor with ECOA1 and blood enzyme, but what do you do with the inserts to actually ensure they fit into the factor, because they have to be digested as well?
Yeah, so that's actually a very good question. So the reason why we clone these or digest both the cDNA inserts and our phase display vectors with the same restriction enzymes is that, maybe I can show it over here. You see that if you have your vector, this is the vector sequence, and if you go to ECOA1, and ECOA1 cuts after the G before AATTC. So this is our vector, which is then cleaved open after the G over here. And if we then have our cDNA fragments, they will also be cleaved at the G, after the G, AATTC. And in this case, you just join these together.
Okay, and you get your cDNA fragments by beforehand digesting it with those enzymes?
Yeah.
Okay.
Yeah. So that's what I actually show over here. So we prepare first, maybe that's... maybe I went very fast to this. We first make our cDNAs in a plasmid factor, and when these are present, then we just cleave these cDNAs out of our plasmid factor and then convert it in a phase display factor. So, and how do we do this? We prepare the DNA from the plasmid factor. This is what then we digested, and these are the digested fragments. So you see all the smears over here, but these smears correspond to all the different. And then we isolate it just out of the shell. That's why I really wanted to show you how we do this, because we always speak about cloning, but some people they don't know how do we do this in practice. So we really need to isolate this out of the shell. Other questions? So after the construction of this phase display library, we can start our serological antigen selection procedure. So how is this working? We first, of course, prepare our phages from our RNA phase display library. So all the different phages express different antigenic fragments, so proteins, and then we incubate these with the antibodies present in the RA serum. So what is now going to happen? If there are antibodies present in the serum of the patient from RA, can bind to RA-specific targets that will be expressed on phages, there will be a complex formed. You see that there's antibody-antigen interaction over here. Then we fish out the phage antibody complexes. So we fish out only the antibodies that are bound to specific phage targets over here. And then the next step, of course, is that we wash the ones that are not bound, what you see over here, and then we retain the bound phages and we start a novel selection round. So in this way, this allows us to enrich us for binding of specific antibodies to specific antigenic targets of the protein. So we repeat this procedure again. So we prepare again phages that we selected from the first round. Then we incubate these again with the antibodies present in the serum of these RA patients. Again, we will have interaction, and in this way, this leads to enrichment. So this is just another cartoon of the serological antigen procedure. So we start with our phages. We incubate it with serum antibodies. We then form antibody-antigen complexes. We capture the RA antibody binding phage. And how do we do this? We just incubate a plastic tube with an antibody which is directed against the antibodies present in the serum. And here we're looking for IgG responses. So how do we capture these antibodies? By using an antibody against IgG. So this is actually how we capture these complexes. Then we just get rid of the non-bound phage, and then we elute these selected phage, so the ones that have bound over here. We elute these and start another cycle of selection. So by repeating this procedure again and again and again, and in practice we most of the time do this 4 to 5 times, we can selectively enrich for specific antibodies that have bound to their target antigen. So that's the procedure. So selection and amplification are the steps for the SOS technology. So what is the result? Of course, that we have an enrichment of phage clones that display antigens that are recognized by the RA antibodies. And this is again the different phases of the phage display. So we first have exposed our RA cDNA library to patient serum. Then we have recovered the phage that bound to the RA serum. And the next step is, of course, that we produce and evaluate the candidate RA antigen. So how does this work in practice? So this is actually, again, the selection of the cDNA display library on serum. So here we performed 4 rounds of selections. So we did 4 rounds, preparing phage, incubated with antibodies present in the serum. And finally, by performing this 4 times, we finally had an enrichment of specific phage. The titers went up. And we see, how can we then see if we have enrichment of specific clones? We do a kind of fingerprinting. So what do? Is the phage, of course, infect bacteria because they cannot amplify on their own. So you infect bacteria and just plate the bacteria on a plate. And you can then pick single colonies from the plate and then determine the insert in each single colony. If you do this and then incubate this also with a restriction enzyme that just selectively recognizes specific restriction digestion pattern, then you end up with different fingerprints. And what do you see over here? That, for instance, number 3 and 4 are the same, that number 8 and 18 are the same, and that number 10, 14, 16, 17, and 19 are also the same. And this means actually that these clones are selectively enriched. So that's actually how we determine whether our selection procedure has worked. The next step is, of course, that if we have these clones identified, we just pick these enriched clones and do the sequencing analysis. And because we know how we clone these cDNA fragments in our phage display vectors, we can initially do a sequencing and looking at the insert, because we know the gene 6 sequence, we know the linker sequence, and then we know exactly at what site our insert starts. What do? We just sequence the insert and then, of course, do a blast search to really look to which target do they correspond. So after identification of the targets, we just want to see whether we have specific immunoreactivity detected there, because that's the way or the basis on how these clones were selected. So how do we then go on? We have selected some immunoglobulin reactor cDNAs, and these are some targets that we sequence over here. And the next step is then to look for immunoreactivity to a number of RA and control sera in order to see whether the antibody response is specific to RA patients and not present in controls. And we do this by in the first stage by a phage ELISA, and later on we develop protein ELISA and peptide ELISA. So this is just the procedure how we did it in the laboratory. To perform our study, we really wanted to have new biomarkers for patients with an early disease and patients which were currently negative for both serological markers in the clinic, so rheumatoid factor and ACPA. So what did we do? After we've created our RA display library, we then performed our selection procedure with serum from patients with an early disease, and in another tube we did the SAS procedure with serum from patients with rheumatoid factor negative and ACPA negative serum. And what was then the plan? Of course, we performed our serological antigen selection procedure. We selected for RA antibody binding phage, and the next step is, of course, when we identified some enriched clones, when we sequenced them, we then did a high throughput screening of the selected phage by testing a number of sera from RA patients and controls to look for antibody reactivity against these targets that we identified. So this is just characteristics of the RA patients that we used for affinity selections. So we had one pool of patients with early disease. So here you see the disease duration in months indicated. And here you have the patients which are still negative. So these are the long disease duration, but they're negative for both serological markers, rheumatoid factor and ACPA. So after this procedure, we identified actually 14 enriched novel antibody targets. And what's really important to say that we first renamed these markers in University Hasselt with the number of the markers that we identified. And then we looked at antibody responses in a group of RA patients that we collected here at the university. And we then looked at how many of our patients do show antibody responses against the novel markers that we identified. So this was our first study that we did. We did all this by phage ELISA. The next step is to look for antibody reactivity in the validation cohort. This was a serum that we obtained from a clinic in Nijmegen. So we blindly contained a serum from RA patients, and we also tested for antibody responses against the targets that we identified. Of course, when you want to look for new markers, we also need to look for antibody reactivity in control groups. So these are healthy control groups, but also, for instance, patients with mechanical joint complaints. So they come to the neurologist, they say, I have pain in my knee, but the neurologist needs to decide, is this patient an RA patient or not? And it seems that if it's not, then these people are called patients with mechanical joint complaints. And of course, it's also important that we also need to test for antibody reactivity in some other patients with inflammatory rheumatic diseases, because I told you that RA is a group of diseases, so that it can also be antibodies found in patients with inflammatory rheumatic diseases as well, which are not clearly RA patients. And then we also test for antibody reactivity in other disease group, which is patients with osteoarthritis, which is different from rheumatoid arthritis. And what did we see actually? We first, in our initial cohort, we had approximately almost 60% of the patients which showed reactivity to at least one of the 14 markers that we tested. We kind of confirmed this in our validation cohort. We did not see antibody reactivity in healthy controls and patients with mechanical joint complaints, and did see some reactivity in patients with inflammatory rheumatic diseases, and also in one patient with osteoarthritis. So these are just, again, the sensitivities and the specificities for the single markers indicated over here. So you see that for some of the markers there is very low sensitivity with high specificity, and for the other markers there is somewhat lesser specificity, but a very high sensitivity. So the next step is, of course, to look for the most promising markers out of the pool. So if we just combined the autoantibody panel with the 11 RA-specific antibodies, so where there was no antibody reactivity present in the control group, so indicated in here, we had a sensitivity of 37% and a specificity of 100%. And when we then included all the 14 markers that we identified, then we had a sensitivity of 54% and a specificity of 9%. So that was our initial finding. What we actually wanted to know is how many of our patients that we identified are seronegative for the current serological markers, because we really want to have markers that can add something in the clinic, and that's what we did. We just looked in all the different patient groups that we had, and we selected or determined how many of our RA patients are positive for both rheumatoid factor and ACPA, are negative for both, are positive for one of both. And then we looked for immunoreactivity against all the different markers over here. And what you see, and this is more interesting, of course, in the group of patients that were currently seronegative based on the clinical test, we did see that half of the patients did show reactivity to at least one of the markers that we identified. So if you then look, or just have a look at another view, we just see the presence of the autoantibodies in the seronegative patients. So here you see the indication. This was the patients positive for both marks, positive for one, and then the seronegative one. And in this group, we looked how many were positive for the panel and negative for the panel. And what you then actually see is that at the end, we could increase the sensitivity of serological testing by almost 20%. So this just depends on whether we just test for the 11 RA-specific markers or whether we test for the 14 RA-associated. So this really indicates that we have some potential markers that can add something to the clinic. But of course, the next step is that we need to do some evaluation. So this is just to summarize this part. So we, of course, looked at when are these antibodies present. Of course, we looked in 10 early RA patients, and we saw again that these patients show antibody reactivity already in an early disease stage, because that's what we want. And we also found the prognostic potential of these antibodies against the marker, because we found an association between positive for these markers and also higher CRP levels. That means that patients have higher inflammation. So it means if you have antibody reactivity against this marker, it also says something about the inflammation potential. So what did we then do is, or what are we now doing, is actually testing antibody reactivity in undifferentiated arthritis patients, and we do a longitudinal follow-up in order to see whether we can also find this antibody reactivity in patients which have not reached the final stage of rheumatoid arthritis, and these patients we call undifferentiated arthritis patients. So which components were bound to these RA antibodies? I'm not going to discuss this in detail, but just to mention you that all the targets that we identified in our technology were novel proteins that were never described in relation to RA. So that's an advantage of the technology. So if you have a broad source of antigen available, you have, of course, you can pick up new antigens. If you compare this, for instance, to protein arrays, when you're looking for antibody reactivity to proteins, the difference is that in the proteins arrays, you only have the known proteins available on the array, while here, when you start with synovial tissue and expression of the cDNA into the proteins, you have all the information present in the synovial tissue. So we did find homology to known proteins that were present in the database, but was also very exciting for us is we also find some homology to peptide sequences, and these peptides, they corresponded to some joint components, so also to some autoantigens that were described already, but also to some pathogens that were suspected to be related to rheumatoid arthritis, but with other people have never proven that they really were the trigger for rheumatoid arthritis. So what does that mean? If you have a certain alignment to these peptide sequences, we call these so-called mimotopes. I will explain this later on. So why did we now find antibody reactivity against these novel markers? We just did a more detailed study on this. We really want to see, why do these patients develop antibody reactivity against these proteins? And therefore, we looked at the expression of these proteins in RA tissue and control tissue. This is just for 3 known proteins that were present, HLA-A, MCM2, and RPS6. And another one, which was number 21, that was like a so-called mimotope, so we don't really know the real in vivo target. So we purified here the antibodies against this peptide out of the patient's sera, and we did then stainings with the serum from these patients. And what you see, if you compare the expression of RA, of the proteins in RA tissue compared to control tissue, that indicates that there is an increased expression of these antigen in the RA tissue. So we may think that the reason why we have antibody reactivity against these targets is maybe that we have overexpression of these proteins in the tissue. This leads to immune tolerance breaking, and then in that way we can have antibody response. That's our hypothesis. We still prove it. So I just want to make that clear. So after doing this ELISA on the phages, so we go to the next step, is to do the validation phase. So we really want to have a test that you can also apply in the clinic, because not all clinical labs can work with filamentous phages to look for antibody reactivity. So we need to develop protein and peptide ELISAs. So what is the difference between a phage ELISA and, of course, a protein or a peptide ELISA? Here, we first need to prepare the phages. This is for the number 21. We then look for antibody reactivity against the phage. We capture this by an anti-phage capture antibody, and then detect the signal by using an antibody against IgG responses over here. So we have phage involved here, and it's quite labor intensive because we need to produce phages. How do we do the peptide ELISA? Of course, it's much more straightforward. You just go to your ELISA plate with your peptide corresponding to 21. You then look for a screen with a serum or plasma from the patient, and then detect with an IgG. So it's much more steady, and it's easily reproducible. And this is what we had to do, because we submitted our paper based on our phage ELISA results, and then the reviewer said, oh, it's very nice and clear, but I really want you to confirm it using recombinant proteins or peptide ELISA. And that's what we actually did. We just selected a number of the markers, so you see number 1, 2, 7, 8, 9, 11, 13, 14, etc. And we then tested patients which were positive in the phage ELISA and negative in the phage ELISA, and tested these in the protein and the peptide ELISAs. And here we could confirm that the antibody response that was really directed against that peptide or protein that we identified. So that was a confirmation of the antigen specificity. We also had to convince the reviewers about a specific antibody reaction in another way. So how did we do this? We performed some competition assays. So this is just our standard phage ELISA. So we just capture the phage. Then we come with the phage with the marker. We look for antibody reactivity against the marker and detect the antibody reactivity by an antigen IgG. So how is the competition assay working? We first do a pre-incubation with plasma antibodies against a synthetic peptide. So that's a peptide that we synthesize, and the sequence corresponds exactly to the peptide as it is displayed on the phage. So what is then actually happening? When there is binding in the pre-incubation of antibodies present in the serum to the synthetic peptide, when we then measure this in the phage ELISA, we will have a decrease in the signal at the end. And that's the competition experiment that we did. So finally, we will not have binding because already in the pre-incubation step we had interaction with plasma antibodies and the peptides. And this is what we proved. I'm not going to discuss it in detail, but we also had to convince the reviewers that the antibody reaction against the targets that we found was really the target that we claimed that was the target. And we also had to confirm this. So what did we do? We just added a different concentration of the peptide in the pre-incubation. And the more peptide we, of course, added in the pre-incubation step, later on, the more competition we had at the end. And this is what we could prove for a number of the markers that we identified. Then, of course, we are basic researchers, and we had identified number 21, and it was the one that was kind of highly sensitive, or the most promising one in our group, because we found it in a significant number of RA patients, and the specificity was also very good because we had a 95% specificity. And we really wanted to do some more research on that. And one thing that we did is to look for antibody reactivity. Is this antibody reactivity against this marker present in all subpopulations? And it seems that also patients that were currently negative for rheumatoid factor and ACPA did show antibody reactivity against this marker. So we are doing some more research on this in the market currently. We are looking at the expression of this marker in the synovial tissue, and I already showed you an example. But the most intriguing thing is that we don't know the exact real in vivo target of this marker, because it's a nemotope. So what is a nemotope? It's actually an epitope which mimics the structure of a real in vivo antigen. So that means when we had the sequence of this 21 marker, the peptide sequence, when we did a search in the databases for the proteins, we did not find a hit for 100%. So it could be that the marker that we identified, I can show you. So if this is our 21 marker, that is expressed like this on the page, and that you just have a few amino acids that determine the epitope binding. So that's something we are now investigating. So we really want to know the real in vivo target of our 21 antigen. So how do we do this? What are we doing? We collect a lot of serum from patients that were positive for this marker 21. We collect the blood. We isolate, of course, the antibodies out of the plasma, and we then do an incubation with synovial tissue that we obtained from patients, and then by proteomics technologies and mass spectrometry, we really want to find the real in vivo target, and that's work that's currently going on. So to conclude, I think I showed you that using the study, we identified at this moment 11 RA specific and 3 RA associated antibody markers. And it's really important that we did find antibody reactivity against these markers in patients that are currently seronegative. So if we can include these markers in the diagnostic testing for rheumatoid and ACPA, we can actually increase the number of patients that will be diagnosed. And I can tell you that we're now collaborating with a big American diagnostic company. They're actually validating our markers in order to incorporate our markers in their diagnostic kit, because they sell the kit to all the clinical labs, and they're currently testing the potential of our markers really in these patients with the early disease. And of course, we are doing some more work on the value of these markers, not only from a diagnostic point of view, because we really want to know what is the real in vivo target. So we already did some stainings with antibodies against this marker, so we found increased expression of these antigens in our RA synovial tissue. And we also really want to see whether the expression, for instance, is different in patients which are negative or positive for ACPA or ACCP. So the number 21 is, of course, one of our favorites, but we do see antibody activity in 1 out of 3 RA patients, and we also see it present in all RA subpopulation, but it's also important that we see it in seronegative patients. We did show an increased expression of the protein in some synovial tissues, and what I didn't mention in this presentation, but we also did some animal studies. already where we induced the CI, rheumatoid arthritis, in an animal model. And at the day of the clinical symptoms, we do a passive transfer of the antibodies that purify out of the serum from these patients. And it seems that if we then do this transfer, these animals have a much worse disease outcome. So we're repeating this experiment, which also says that if you have these antibodies present in your serum, it also says something about prognosis potential. So this is just what we're doing. I just mentioned you already that we're collaborating with a diagnostic company in order to validate the diagnostic potential of the markers. But we're also doing a longitudinal screening of patients with undifferentiated arthritis. So therefore, we're collaborating with a group in Leiden. They have a group of 1,000 patients that they followed for years already. And they have follow-up of 7 years of the patients. And we're now testing these samples initially that were taken initially, and then the follow-up, and see whether we can already look for antibody activity in the very, very early stages of the disease. And of course, as I mentioned to you, we are doing the analysis of the biological relevance in order to know the role of these markers in disease pathogenesis. And this is really what I would like to see.